Okay, we were talking about DNA replication, and just to take a couple of steps back here, talked about DNA replication. Okay, for DNA replication to occur, we need what we call our parent double-stranded DNA. Both strands are simply DNA polynucleotide strands. Okay? Both strands serve as templates to synthesize new daughter strands. By the end of DNA replication, we end up with two identical molecules of double-stranded DNA, but each molecule consists of the template parent single-stranded DNA that remains chemically bonded to the new daughter strand. Okay? We talked about how DNA replication is referred to as a semi-conservative process. I believe I briefly mentioned the enzymes where we left off, and this is where I'm going to pick up on. Talked about DNA replication. If it's prokaryotic DNA, it's circular, but if it's linear DNA, to where it has open ends here, as I show you here, that's symbolic of what you see with eukaryotic DNA, such as the DNA in our cells. It's described as linear. And in eukaryotic DNA, okay, DNA replication doesn't begin at one site. It begins at multiple sites, and these sites are called the origin of replication. They're known simply as the ORI, okay? Origin meaning the beginning. This is where DNA replication begins. Well, what I'm going to do now is add in the enzymes. Okay. The enzyme that is responsible for scanning the parent double-stranded DNA and locating this site called the ORI, or origin of replication. It's called DNA helicase. Okay. DNA helicase is responsible for locating and binding to a site along the parent double-stranded DNA called the origin of replication. Once it binds to this site called the ORI, okay, that's located along the parent double-stranded DNA, down here it says its job is to then unwind and separate the double-stranded parent DNA at this origin of replication, okay, resulting in two separate parent single strands of DNA. However, DNA helicase, which they're showing you here in green, okay, initially unwinds only a segment of the parent double-stranded DNA, therefore creating a bubble. What did we call that bubble? What was that bubble called created here? I'm telling you, the enzyme DNA helicase creates this bubble, but what do we call this bubble that was created? It has to do with the process. Not a rhetorical question, everybody. What do we call this bubble that's first created? I will test you through chapter 12 whether we cover it all or not. That bubble is simply called the what, everybody? The replication bubble. DNA helicase creates this replication bubble. And on each end of the replication bubble, we have replication forks. Okay? Now, once DNA helicase creates this replication bubble, what will happen is it's not one single enzyme called DNA helicase. It's actually a group of them with all the same name, DNA helicases. Okay? And what will happen is once that replication bubble is created, a DNA helicase will slide towards this replication fork. Another DNA helicase will slide towards this replication fork. Here they're showing you a DNA helicase that slid towards this end of the replication bubble that we call the replication fork, where single-stranded parent DNA meets double-stranded parent DNA. This is where they're showing you DNA helicase, because DNA helicase's job is not complete once it's created this replication bubble. Okay, remember, at the end of DNA replication, the double-stranded parent DNA has to be completely unwound and separated. Therefore, DNA helicase, once it creates a replication bubble, they'll slide down to the replication forks and continue to unwind and separate the parent double-stranded DNA. So this replication bubble will get longer, or it will elongate. What happens though once this replication bubble is created? We then have our next enzyme coming in. It's called DNA polymerase. Don't worry about the 1 and the 3. There are different forms. When they're Roman numerals, they're talking about DNA polymerases. In prokaryotes, for eukaryotes, the DNA polymerases are given Greek letters, such as alpha, beta, gamma, and delta. But I just want you to know them as DNA polymerase. DNA polymerase goes by the abbreviation DNA pol. So the pol stands for polymerase, but it's an enzyme. Remember, enzymes get names that often end in ASE. DNA polymerase, okay, it'll come in, and its job, as they're showing you right here, here's a DNA polymerase here, here's another one here, here's another one here. The job of DNA polymerase, well, its job is to synthesize the new daughter strand. Except it takes multiple enzymes called DNA polymerase, and these enzymes will use both the forward parent strand and the reverse parent strand. Both DNA strands will be used as templates for synthesis of the new daughter strand. You have to remember this because we're going to go over a second process called gene expression. Okay, and it's going to be a little different, or a lot different. So in this case, both parent strands serve as templates for synthesis of the new daughter strands. But if you look here, nowhere in here that I'm circling does it mention what the daughter strand is. Well, the daughter strand is a light blue color. Okay, ignore the red. There's more to the story I'm not going to test on, but the light blue color is the new daughter strand. What was discovered to happen is once DNA helicase creates this replication bubble at the origin of replication, the enzymes called DNA polymerase will come in, and one will bind up here on this parent single strand, which is the forward strand, 5' to 3', and another DNA polymerase will bind to the opposite parent strand, or the reverse strand, okay, which going to from our right to our left is 5' to 3'. And what will happen is each enzyme will begin to synthesize a new daughter strand, okay, in the light blue color, towards the replication forks. And they'll do it continuously. However, as I said, DNA helicase here, once it creates the replication bubble, it's not through. Okay? DNA helicase enzymes will move to opposite ends of the replication, where the replication forks are, like they're showing you here, and they will continue to unwind and separate DNA. Some teachers are saying unzip. I don't like to say that, but that's what they're showing you here. But unwind and separate. Well, the problem with that is, look at the strand up here, which is the forward strand. Say where the red dot is, DNA polymerase bound to the origin of replication on this parent single forward strand, and it started to synthesize new daughter DNA towards the replication fork continuously. But when it turns around and looks back, what it didn't see is that one of these enzymes, DNA helicase, was also on this side, continuing to unwind and separate DNA. So now what happens is we have additional parent single-stranded DNA that hasn't been replicated. So additional DNA polymerases have to keep coming in to start synthesizing a new daughter strand to help out with this enzyme. A second DNA polymerase will come in, start to synthesize new DNA, but helicase continues to unwind and separate DNA. So another one jumps in, start to synthesize, looks behind it, more single-stranded DNA is present, so another DNA polymerase comes in. So what happens along the forward strand here, 5' to 3', is we have some DNA continuously made towards the replication fork, but some of it is in fragments moving towards this replication fork. The same thing is happening along the reverse strand. Initially, the new daughter strand is made continuously using the reverse parent strand as a template, but again, this helicase over here is continuing to unwind and separate DNA at the replication fork, moving it. So again, additional polymerases. So what they normally do then, okay, is they'll put a box dividing the replication bubble in half, which to me never made sense in genetics. Okay, it never did. When they went over this, I was like, well, what the hell are you saying? So the way they do this is they separate the replication bubble in half to simply point out, okay, that when you're in a replication bubble, okay, along one of the two parent strands, you will see new daughter DNA being made towards the replication fork without any separation. It's just made continuously. When that daughter DNA is made continuously going towards the replication fork, we call that the leading strand. Okay, it's made continuously towards the replication fork, but you have to start in the middle of the bubble to point that out. Okay. Coming directly across, what is happening is that the other daughter strand using the reverse parent strand, it's being made towards the opposite replication fork over here. And if you cut this replication and bubble in half, okay, and look at the new daughter strand being made directly across from this leading daughter strand, it's being made in fragments towards the opposite replication fork. They call this strand the lagging strand, which they show you is going on down here. Okay, so the way it works, all right, is when we take the replication bubble in order to explain these two strands, they'll cut the replication bubble in half like this, just to explain the difference in the appearance of the two strands. Listen carefully. When you hear the terms leading and lagging, I am talking about the daughter DNA strands, please. Please note, so if you hear leading strand or lagging strand, we are talking about the new DNA that is being synthesized, which is called daughter DNA. So it's showing you here, if it's a lagging strand, we start from the middle of the bubble, we go towards the replication fork, and you see there are no breaks. It's just showing you the light blue representing the daughter strand, meaning it's made continuously. If we start in the middle of the replication bubble and we go directly across from the leading strand, we find our lagging strand on the opposite parent strand. But notice the arrows are pointing away from the replication fork because this daughter strand is being made towards this replication fork. But when we cut the bubble in half, we show this other daughter strand is being made in fragments or discontinuously. Each of these daughter DNA fragments is called an Okazaki fragment. The thing is this, we don't want daughter DNA that's made discontinuously or in fragments. If you look down here at these two molecules, notice the daughter DNA in gold, there are no fragments. It's made continuously. So how does the cell fix this problem with the lagging strand? With that part of the daughter DNA called the lagging strands? Well, it has to do with this enzyme, DNA ligase. The job of DNA ligase, okay, is to covalently bond the fragments of DNA together. In other words, if you recall, I said this little strap part here, not the teeth, but the little strap part here. What does that strap part represent, everybody? It doesn't matter if it's in dark blue, it doesn't matter if it's in light blue. What does this solid little strap, not the little teeth that are connected, but what do these solid little straps represent? What type of backbone? Give y'all one more chance on this question. If not, I'm just going to ramble on as quick as I can. Anybody? All right, this represents a sugar phosphate backbone. So what DNA ligase is doing is it's looking for nicks. We call these nicks. Nicks are missing covalent bonds in the sugar phosphate backbone of a DNA polynucleotide. Okay, so what DNA ligase does is it fixes nicks, okay, or missing covalent bonds between these Okazaki fragments of the lagging strand. It will form these covalent bonds so that in the end, the sugar phosphate backbone of the new daughter strand is continuous so that there are no breaks. So DNA ligase will come in and it'll focus on the new daughter strands highlighted here in gold, and anywhere it sees a covalent bond missing in that sugar phosphate backbone, it will create that covalent bond so that now we have one continuous daughter DNA polynucleotide strand. And these will remain base paired with the parent strands. So we go from one molecule of double-stranded DNA to two identical molecules of double-stranded DNA. That's DNA replication. Well, that's DNA replication, okay? And the purpose of DNA is to store genetic information. That's the way we describe it. Well, That's the purpose of the second process. We talk about the central dogma, so let me see if I can go back to that. Central dogma. Dogma means belief. What's at the center of that belief? I said before is DNA. Okay. DNA is at the center of a process called DNA replication. We take parent double-stranded DNA and we replicate it, and we end up with two molecules of double-stranded DNA that are identical. Okay. Why is it important? When one cell divides into two cells, we want to make sure that both cells get identical copies of that double-stranded DNA, because that double-stranded DNA contains genetic information in the form of genes. Okay. What's the importance of this genetic information in the form of genes? Well, these genes code for proteins. Okay. And what did I say proteins do in a cell? These proteins do the work. As a matter of fact, I just told, when you go over DNA replication, what is actually doing the work for a process called DNA replication? It's proteins. Proteins that function as enzymes. DNA helicase, DNA polymerase, DNA ligase. They're all proteins that do work. One type of work they do is a process called DNA replication. Another type of work they do, well, is called gene expression. Right? So DNA replication has to do with taking one molecule of double-stranded DNA and replicating it to where we have two identical molecules. Why is that important? Because genetic information is stored in DNA in the form of genes. What's the importance of genes? Well, genes code for proteins that do the work in your cells. So genes, simply put, you can think of a gene as a protein recipe. And this is what we're going to get into next, the process called gene expression. Now, some people will call gene expression protein synthesis, but that's not correct. Okay, because protein synthesis doesn't make up all of gene expression. Okay? So listen carefully. When we talk about gene expression, this is where we now introduce RNA. Okay. The way it works for gene expression, first of all, is it's a two-step process. The first step is called transcription. The second step is called translation. Okay. Transcription has to do with making a copy of that genetic information stored in a gene, making a copy of it, transcribing it, but in the form of RNA. Okay. In the case of eukaryotic cells have a nucleus which stores DNA. So it's very important, okay, copy of that genetic information in the form of a gene be made, or that it be transcribed in the form of RNA, so that it's easier to transport to where it needs to be taken for translation. So in eukaryotic cells, transcription occurs in the nucleus. Translation occurs out in the cytoplasm, specifically at non-membranous organelles called ribosomes. So it's just a lot easier. Instead of trying to drag the DNA out of the nucleus, which is going to destroy the cell, just transcribe it, which means make a copy of it and transport it to the correct area in the cytoplasm, which will be the ribosomes. Then it'll be the job of the ribosomes, with some help, to perform translation. Okay, so it's a two-step process, transcription and translation. When we go over transcription, there's an enzyme that's the star of the show called RNA polymerase. We will talk about how it's responsible for transcription. When we go over translation, there are more working parts, but it all occurs at the ribosome. But we have to talk about not just messenger, or not just RNA, but three types of RNA. Okay. Now, for gene expression, it varies a little bit. In prokaryotes, these two steps, transcription and translation, can overlap. Okay. When we talk about transcription, we're going to focus on an enzyme called RNA polymerase. It is responsible for the entire process, the entire first step called transcription. And when RNA polymerase performs transcription, it doesn't just make RNA. It makes a type of RNA known as messenger RNA, or mRNA. It's called messenger RNA because, well, the message it's carrying is the genetic information for how to make a protein. But it'll be up to the ribosomes to take that genetic information in mRNA and translate it into the protein that needs to be made. But in prokaryotes, what it happens is that transcription, which is here at the level of DNA, where we go from DNA to RNA, okay, after transcription has occurred for a certain period of time and the mRNA is long enough, the second step can actually begin, which is translation. Now, I told everybody you needed to go back to chapter 3 and go over nucleic acids and proteins. Why? Because it doesn't say protein on this figure, it says polypeptide. If you see polypeptide, we're talking about proteins. Okay, so here they're showing you in prokaryotes transcription up here and translation here at the ribosomes. Okay, they can both happen at the same time. Okay, not in the beginning, but there's a point where they can overlap. All right, but transcription will always begin first in a prokaryote. Then there's a point where transcription and translation will overlap, and then it'll finish with translation. And that can't happen because DNA is stored in the nucleus. Transcription has to start first and be completed so that the mRNA can be transported out into the cytoplasm to be translated. So in eukaryotes, the two processes do not overlap. They're completely separate. Now, sometimes one gene, okay, contains the genetic information to make one protein that is an active protein that goes out and does the work. However, what scientists discovered, okay, is that there are proteins that do work that are complex proteins that are coded for by more than one gene. An example of that is hemoglobin. Okay, there are actually two genes responsible for forming the majority of a molecule known as hemoglobin. One of the genes, its loci is on chromosome 16, the alpha chain gene. The other gene, the beta chain gene, its loci is on chromosome 11. And what scientists discovered is that when gene expression occurs involving these two genes, it doesn't result in what they call proteins or active proteins. It results in what they call protein subunits. Okay. What'll happen then, okay, is in this case, what they mean by protein subunits is that the alpha chain gene codes for a polypeptide that folds into what we call a protein subunit that will then complex together not only with other alpha proteins, alpha chain proteins, but it'll also complex together with protein subunits coded for by this other gene located on a different pair of chromosomes, chromosome 11. This chromosome has a gene called the beta gene, which codes not for an active protein, but for what we call a protein subunit that will then complex with another beta protein subunit that will then complex with these different protein subunits. So in other words, we end up with a complex protein, okay, formed by 4 protein subunits that come together. Okay, we have to have all 4 of these come together to get our functional hemoglobin molecule. So sometimes a gene codes for a protein that is an active protein that goes out and does work. Sometimes a gene codes for a protein subunit that has to attach or complex with other protein subunits to then form an active protein. Now, the first step, we're going to go over transcription. Transcription is described as a 3-step process. Now, in your notes, though, I think I itemized it and listed what was needed. And then under RNA polymerase is where I go over these 3 steps, but I don't have them standing out, but I have them on the PowerPoint. Okay. First of all, for transcription to occur, all right, we need the enzyme RNA polymerase, okay, and we need our DNA. But that DNA we need not only needs to consist of, not only do we need the enzyme and that DNA, but the DNA not only needs to contain the genetic information in the form of a gene, but that gene has to be bracketed by 2 other types or regions of DNA. Okay? One of these regions of DNA is known as the promoter. The promoter is always located just before the gene, which they show you here in green. The other region of DNA that is needed, well, it's bookending the gene. It's located on the opposite end of the gene. This is your terminator or terminator DNA. So these are called promoter DNA, terminator DNA, or promoter sequence, or terminator sequence, or just promoter and terminator. But these two are regions of DNA that bookend or bracket the gene. Okay. What else we need are RNA nucleotides, because what RNA polymerase is going to do is it's going to synthesize RNA. Okay. And not just any RNA, but as I said before, it's actually going to synthesize a type of RNA, one of three types, called mRNA. So these are the working parts that we need. Okay. Now, RNA polymerase, unlike DNA polymerase, it does pretty much everything on its own as long as it has access to the DNA. Now, what RNA polymerase will do, okay, is when it performs transcription, it'll do it in 3 steps. The first step is RNA polymerase first has to locate the promoter to initiate transcription. The promoter is a region of DNA just before the gene, and this promoter helps RNA polymerase locate the gene. And not just locate the gene, but locate the start of the gene. In other words, help RNA polymerase not only locate the gene, but locate the beginning of the gene so it knows where to start transcription. Okay? What will happen then is once RNA polymerase locates the promoter, it will then unwind and separate the parent double-stranded DNA. Okay? Creating okay, I'm not going to spell all this out. What I'm trying to spell here is shoot, I knew I was going to do that. Oh, I didn't curse though. Okay. Creating and I told myself before, I'll look at my notes to try to make sure I'm trying to say something halfway coherent, decent. I'm not going to spell the rest of this out, but what I'm trying to write out is transcription bubble. Sorry. Tell myself, don't confuse the bubbles. Replication bubble happens during DNA replication. Transcription bubble happens during transcription, the first of 2 steps. So that was an ugly E. Should be an E. All right, I'm just making it worse. So listen carefully. Once RNA polymerase locates the promoter, it will bind to the promoter, slide over to the beginning of the gene, unwind and separate the parent double-stranded DNA, creating a transcription bubble. Once this transcription bubble is created, okay, that's the end of the first step of transcription, called initiation. The catch is this, okay, RNA polymerase does not use both parent strands, okay, that store the genetic information. RNA polymerase does not use both parent strands as a template to make RNA. That's what this slide is for. What will happen, okay, is that RNA polymerase down here, that RNA polymerase, when it locates the promoter and slides over to the gene and unwinds the parent double-stranded DNA, it then identifies which strand is the reverse strand. For transcription, the first step in gene expression, only the reverse strand serves as a template to synthesize mRNA. Okay? So this is different from DNA replication. Only the reverse strand is used as a template to synthesize mRNA by RNA polymerase during transcription. Okay? Now, if you notice here, they're showing you when it uses that reverse DNA template strand to synthesize mRNA, RNA polymerase is going to synthesize mRNA, notice the direction, in a forward direction. Okay? You got to go back and look at the notes I gave you. Okay? 5' to 3' from our left to our right, that means forward. 3' to 5' from our left to our right, that means reverse. Notice they're showing you something here. Okay? But once it's created this transcription bubble, and it's located which strand is the reverse, which is highlighted here. You can see blue parent DNA, but you can see red RNA where it's initiating transcription. Okay, what they're showing you is RNA polymerase is located which of the two strands is a reverse. That's going to serve as a template strand to begin the second step of transcription, which is elongation. Okay, elongation, simply put, elongation is where RNA polymerase synthesizes mRNA. That's elongation. So it should be mRNA. Okay. So elongation, this is a process or step where RNA polymerase simply synthesizes mRNA using the reverse parent strand as a template. Now, during elongation, the transcription bubble does not grow longer or does not elongate. Instead, the transcription bubble moves along with RNA polymerase. And what I mean by that is what scientists discovered. The green represents the promoter. Is once RNA polymerase slides over to the beginning of the gene after it's located the promoter, it'll unwind and separate the parent DNA, locate which one is a reverse strand, and begin to use that as a template to synthesize mRNA. And as it synthesizes mRNA, RNA polymerase will move from our left to our right. And as it moves downstream, as we call it, it'll continue to unwind and separate parent DNA using the reverse strand as a template. But the DNA that's already used for elongation to make synthesize mRNA, RNA polymerase will let go of that parent DNA and it will automatically chemically bond with each other, reassuming that double-stranded helix. So what they discovered is that transcription bubble, you will only find it where RNA polymerase is present because RNA polymerase is creating it. RNA polymerase continues to move downstream. It will unwind and separate parent DNA only where it's bound to the parent DNA. Any parent DNA where it's already used a reverse strand to synthesize mRNA, it will let it go and it will automatically reassume its normal shape. So in transcription, what I'm saying is during transcription, the parent double-stranded DNA should never be permanently separated. It's only temporarily separated to allow for synthesis of mRNA. RNA polymerase will continue to unwind or move this transcription bubble downstream, synthesizing mRNA. Now, what they're showing you next is they've taken this and they've magnified it. Remember I said you have to have RNA nucleotides, okay? This big old guitar pick looking thing is our RNA polymerase, and here's our reverse strand. So here's everything we need I gave you, I believe, at the end of page 1. But notice something. They're showing you in dark blue is the reverse strand, okay? Here's our transcription bubble. And what they're trying to show you, okay, is one, the mRNA is being made in a forward direction, 5' in the direction of 3', using the reverse parent DNA strand. The other thing is this, okay, which RNA nucleotides does RNA polymerase know to choose? Remember, there's 4 different RNA nucleotides based on the 4 bases, A, G, C, U. Well, what RNA polymerase does is it'll look at a DNA nucleotide that has thymine, and it will choose an RNA nucleotide that has adenine. If you recall, we talked about base pairing. Okay, so what will happen is adenine base pairs with thymine. It'll go over to the next DNA nucleotide on the reverse strand. It sees a DNA nucleotide with the base adenine. It will choose an RNA nucleotide with the base uracil, because remember, we don't find thymine as a base in RNA nucleotides. Uracil substitutes for thymine, or U substitutes for T. Well, what scientists discovered is that they also base pair together, just like thymine and adenine. And then RNA polymerase will continue to do the same thing. Hey, here are two DNA nucleotides with the bases guanine. It'll choose two RNA nucleotides with a complementary base, cytosine. So the mRNA that's made in the forward direction is the complement of the parent reverse strand. What do I mean by complement? The bases complement each other, except as RNA polymerase is synthesizing or elongating this mRNA, it doesn't leave it attached to the reverse strand. It knocks it off as it's making it to ensure that the parent DNA can rebond with each other, at the same time making sure that the mRNA is free to be transferred. Now, RNA polymerase needs to know not only where to start or initiate transcription, it also needs to know where to stop transcription. Okay? If you're somebody that likes to cook, or you're somebody that likes to build things, but you depend on instructions to do it, then you know if you're being given a copy of the recipe to cook something or the instructions to build something, you know it's important that that copy is accurate, that it begins at the right spot and it ends at the right spot. So what you produce is not something that's a disaster. Okay? Well, same thing here. Just like RNA polymerase needs to know where to initiate transcription, it needs to know where to terminate it. Okay? Well, what helps RNA polymerase terminate transcription has to do with the region of DNA called the terminator. The terminator is always located opposite of the promoter. Or another way to say it is the terminator bookends the other end of the gene. So eventually what happens if you come down here is as RNA polymerase is performing the second step, elongation, synthesizing an ever lengthening or growing mRNA molecule, eventually it'll come across a region of DNA, okay, that tells RNA polymerase you are now at a terminator sequence, or you are now at terminator DNA, therefore it's time to stop transcription. So the terminator sequence or terminator DNA tells RNA polymerase when to stop transcription. What will happen is when RNA polymerase reaches the terminator sequence, okay, the terminator sequence tells RNA polymerase, let go of the mRNA you just synthesized and let go of the parent DNA. Okay, so again, eventually RNA polymerase, as it's performing elongation, synthesizing the ever-growing mRNA, it will eventually reach a stretch of DNA called terminator DNA or terminator sequence. And as it's performing elongation and it reaches this terminator DNA, that terminator DNA tells RNA polymerase, release the mRNA you just synthesized and release the parent DNA. You are through. Stop transcription. Okay? And then RNA polymerase will go do it all over again if it needs to. In the meantime, for the mRNA that's produced, in prokaryotes, the mRNA that is synthesized is immediately translated. That's another reason why in prokaryotes, transcription and translation, those two steps of gene expression can overlap at some point. But in eukaryotic cells, not only does the nucleus prevent transcription and translation from overlapping in gene expression, but also in eukaryotes, RNA polymerase initially synthesizes what is called pre-mRNA. This mRNA is not ready to be translated. This only occurs in eukaryotes. Okay? This pre-mRNA has to undergo what we call post-transcriptional modifications. Okay? The modifications simply are the addition of a 5' cap highlighted in yellow, and the addition of a 3' poly A tail. Are added to the mRNA molecule. Okay, so notice they don't show any teeth coming off, okay, here, but they're just not trying to make it stand out. So this is only for eukaryotes. The initial RNA that is synthesized by RNA polymerase, it is called pre-mRNA. It has to be post-transcriptionally modified. A molecule called a cap is added to the 5' end, thus we call it a 5' cap. And then a small stretch of RNA polynucleotide, where every RNA nucleotide has the base adenine, is added to the 3' end. Therefore, they call this small RNA polynucleotide, where all the RNA nucleotides have the base adenine, they simply refer to it as a poly A tail. Once these two modifications have occurred, okay, we now have our final product in eukaryotes that is mRNA readily translated. Okay, so this is an intermediate step post-transcriptional modification. Okay, it doesn't fall under transcription, it doesn't fall under translation, it's an in-between step, intermediate step for eukaryotes only. Okay, what scientists discovered is that a single mRNA molecule can actually undergo the second step of gene expression, translation, multiple times at the same time. So what I'm showing you here is the second step, translation. What scientists discovered is that once mRNA is ready to be translated, well, first of all, translation occurs at non-membranous organelles called ribosomes. Now, does the ribosome do everything itself? No. But what they discovered is translation occurs at non-membranous organelles called ribosomes. What else they discovered is that one molecule of mRNA, which they're showing you here, can be translated simultaneously by more than one ribosome, resulting in multiple proteins. So in other words, many active proteins can be produced off one molecule of mRNA, but all these active proteins are the same identical protein. Okay? Now, second step we're going to focus on is translation. Okay, but in order to focus on translation, you have to start with the mRNA. What scientists discovered, first of all, is that for translation to occur, okay, it not only has to occur at the ribosome, but it requires a total of types of RNA. mRNA, and then the other two types of RNA are called tRNA and rRNA. So I'm going to go over these, okay, each one at a time. Then I will go over the ribosome, and then we'll put it all together and go over translation, the 3 steps. And first of all, for the 3 types of RNA, we start with mRNA, and that was a horrible M. Okay, so this represents mRNA over here. The M in mRNA stands for messenger. The message it's carrying, well, it's the genetic information on how to make a protein that a cell needs to do work. What scientists discovered is that genetic information carried in mRNA is coded. It's in the form of a code. So that when a ribosome, okay, is performing the main part of, or performing translation with some help, they discovered the ribosome, okay, doesn't read the genetic information stored in or carried in mRNA one base at a time. The ribosome knows to read the genetic information in mRNA 3 bases at a time. In other words, what they discovered is that the ribosome knows the genetic information is coded. It's in a code. So when a ribosome performs translation, it knows that the genetic information being carried in mRNA is in a coded format. It knows that this coded format is in the form of what we call base triplets called codons. And what a ribosome will do, okay, is it'll read one codon at a time, not one base at a time of each RNA nucleotide, but one codon at a time. In other words, it'll read this base triplet, then it'll shift down, and it'll read this base triplet, it'll read this. The reason for that is because it was discovered each codon codes for any one of 20 amino acids in the human body. So what they're trying to show you here is that during translation, okay, the genetic information in mRNA is coded. It comes in the form of base triplets called codons. Each codon codes for a specific amino acid. Thus, they show you 3 different shapes and then another shape that's a square. So they're trying to show you that this base triplet, known as a codon, codes for one type of amino acid, but this second codon here codes for a different amino acid. This third codon here codes for another different amino acid. And then if you look at this codon, it reads the same as this codon. Thus, we put a square just like up here. Okay. So the genetic information being transferred by messenger RNA, okay, is coded, and it comes in the form of base triplets called codons. Well, what scientists eventually discovered is that there are more codons than there are amino acids in the human body or in cells. 20 different amino acids in our body, but there's way more than 20 codons. Okay, and that's another story we leave for our majors in genetics, our science majors in genetics and biochem. Okay, what I want to show you though is that not only did they discover that there are more codons than there are amino acids, but some amino acids have multiple codons that code for the same amino acid. Example, and I have this in your notes, is tyrosine, known as TYR. There are at least 2 different codons that you can find in a single molecule of mRNA, okay, that code for tyrosine. And whenever a ribosome comes across this codon in mRNA or this codon in mRNA, the ribosome note says, okay, I need the amino acid tyrosine. These are other ones. I'm not going to test you on this one, but serine here and here, this is another one of the 20 amino acids. It has up to 6 different codons that the ribosome knows, hey, that means serine. But if you look here, there are some other codons I do want you to know that are on your notes. Just like for transcription, where RNA polymerase needs a DNA sequence called the promoter, so it knows where to begin transcription. And just like RNA polymerase needs a DNA sequence called the terminator, so it knows when to end transcription, it's similar to what the ribosome needs. The ribosome needs to know, where along the molecule of mRNA do I begin translation? Well, what scientists discovered, no problem, is that built into the mRNA is a codon known as the start codon. And the start codon will always be located closer to the 5' cap. Okay? If you look, there's all these different codons, but they're showing you there's only one base triplet, only one codon that is known as the start codon. That codon is AUG. Okay? AUG is a start codon that codes for the amino acid methionine. Okay? So let me circle that a little differently. So AUG is known as our start codon, and it codes for the amino methionine. And you will find it towards the 5' cap. Okay? Now the ribosome gets some help, but the ribosome knows that when it grabs on, when it binds to mRNA, where it binds to the mRNA depends on where the start codon is. So with some help, the ribosome will end up binding towards the 5' cap, specifically making sure it grabs onto this start codon known as AUG. So imagine you're looking at 3 RNA nucleotides. One has a base A, one has a base U, one has a base G, and it's located right here at the beginning of the red. So that helps the ribosome know where to begin translation along the mRNA. But the ribosome also needs to know, when do I stop translation? Well, that's here. What scientists discovered is that out of all the codons that code for all these different 20 amino acids, there are 3 that do not code for any amino acid. Oh, Jesus. Too much coffee this morning for me. What they found is that there are 3 codons that do not code for any amino acid. These 3 codons are simply known as stop codons. These stop codons are located close to the 3' poly A tail. Okay? Now the way it works is you won't find all 3 stop codons in one single molecule of mRNA. You will find any one out of these 3 here towards the 3' end close to the poly A tail. These 3 stop codons are UAA, UAG, and UGA. When a ribosome is translating the genetic information in the form of codons in mRNA, it knows when to terminate translation when it reaches one of these stop codons. So again, you won't find all 3 stop codons in one molecule of mRNA. It'll either be just this one, or just this one. But the ribosome knows when it encounters one of these stop codons, that tells the ribosome, hey, stop translation, stop synthesizing the protein, you're finished. Okay? So everything is in that molecule of messenger RNA that the ribosome needs to perform translation. Hey, this is where you start. This is where you stop. And this is everything you're going to add to create the protein. But as I said before, it actually requires a total of 3 types of RNA. There's messenger RNA, then there's the second RNA. T stands for transfer. Because this RNA's job is to transfer an amino acid to the ribosome. Okay. Now, so I use cooking shows as an example and stuff, but you might see this what do you call it, mechanic repair shops and everything. You've got the main person that's cooking the meal or that's doing the repairs, and then you have the assistant that's responsible for bringing the necessary supplies. So in this case, transfer RNA, what it does is it helps assist the ribosome in translating the codons that are in mRNA. Okay. In order to help the ribosome do that, all right, tRNAs have their own base triplet, which they're indicating here. Okay. Except their base triplet is not called a codon, it's called an anticodon. Okay. Listen carefully. Transfer RNAs are RNA molecules that on one end have a base triplet called an anticodon. The anticodon complements the codons in mRNA. So again, this base triplet known as the anticodon in tRNA, these tRNA anticodons complement the codons in mRNA except for the stop codons. There are no tRNAs that have an anticodon that complements a stop codon. This prevents tRNAs from accidentally bringing an amino acid to the ribosome when the ribosome is actually supposed to be terminating translation or stopping it. Okay? So on one side we have an anticodon. On the other side of transfer RNA, you will have an amino acid attached. Okay? That's where it gets its name transfer. tRNA is responsible for transferring the correct amino acid to the ribosome. What ensures the tRNA transfers the correct amino acid to the ribosome? The anticodon. And we'll put this all together when we go over the 3 steps. Then there's also RNA known as ribosomal RNA. All I want you to know for ribosomal RNA is ribosomal RNA will complex together with proteins to form our ribosomes. Okay, so again, for ribosomal RNA, all I want you to know is ribosomal RNA will complex or come together and bind with proteins, forming a larger structure called a ribosome. Okay, and that's what I'm not going to test you on this here, but it's just showing you there's different types of ribosomal RNAs that will come together with these proteins, and then there's other ribosomal RNAs that will come together with these proteins to form these two guys. Well, this is more technical than I want you to be. So scratch out these numbers, but keep the subunit. What happens is these, I'm going to abbreviate this. What happens is that some of these ribosomal RNAs will complex or bind to proteins, forming a large ribosomal subunit. And then some of these ribosomal RNAs will complex or bind to proteins, forming a small ribosomal subunit. And then when these two subunits come together, they'll form a fully functional ribosome. Okay, so I cover ribosomal RNA last because it bridges the gap with the last part we need for translation, which is the ribosome. Okay, so I put it here. Ribosomes are formed by ribosomal RNA and proteins coming together to form a large subunit and a small ribosomal subunit. Now listen carefully. The only time these two subunits, the large and the small, come together is during translation. If translation's not occurring, these two will disassociate or break apart. Okay, but when translation's occurring, the large subunit and the small subunit will complex together, creating three sites. One site is called the E site, the other site is called the P site, and the last site is called the A site. Okay, the way these sites work, well, it begins with the A site. The A site, this is what the A site stands for, amino acid. I don't want you to know that. We're going to cheat. Okay, the A and A site, I want you to know the A is standing for access. ACCESS, access. Okay, that's what I want you to know the A for. The P site, that's the second site here. Okay, this is what the P site actually stands for, peptidyl. I don't want you to know it that way. I want you to know the P site. We're going to cheat. I want you to know the P as standing for polypeptide or growing polypeptide. That's how I want you to know the P site. This is where we'll find the growing polypeptide. You can see here a little exit tunnel. The E, well, the E stands for exit, and that's what I want you to know it as. So this one, we're not cheating. I'm giving you the truth. The E is exit. Okay. These three sites, the A site, the P site, and the E site, the way they work is going in this direction. And what moves through these sites? Your tRNAs. Your tRNAs move to three sites. Okay? When a tRNA is bringing, when a tRNA is transferring an amino acid to the ribosome, it will enter the ribosome or access the ribosome at the A site when it transfers the amino acid. What ensures it's transferring the correct amino acid? The tRNA will have an anticodon that complements the mRNA codon in the A site. What will happen then is this tRNA that enters the access site will be shifted by the ribosome over to the P site. In the P site is where you will have a tRNA whose anticodon is still chemically bonded to or base paired to the codon of mRNA. And then on the other end, that tRNA in the P site has that growing polypeptide or growing protein. Then what will happen next is that tRNA in the P site, it's going to have this growing polypeptide removed from it, detached from it. And the ribosome is going to shift that tRNA over to the E site. The E, as I said, stands for exit, and they're showing you here, this is where a tRNA without any amino acid will exit the ribosome because it's done its job. So tRNAs enter into the A site, move to the P site, and then exit without any amino acids through the E site. So this is why they label these three sites. Now, why does the ribosome get credit Okay, for the actual synthesis of the protein or the polypeptide, going into the test, I tend to just say synthesis of the protein. I don't get as technical, but when you see polypeptide, just think protein. Um, the reason why the ribosome gets credit for synthesis of the protein, okay, is because it's responsible for taking the amino acids that tRNA transferred to the ribosome, taking these amino acids and covalently bonding them together. Except we give that covalent bond a special name. Okay? That covalent bond linking one amino acid to the other, we call it a peptide bond. And it's only this bond. Remember, proteins like nucleic acids, carbohydrates, and lipids, they're all organic compounds. They all have carbon backbones, and carbon loves to form single and double covalent bonds. So please listen carefully. The only covalent bond given this special name, peptide bond, is the covalent bond, okay, attaching one amino acid to another amino acid. So it's just this one that's highlighted in gold. That's the only covalent bond called a peptide bond, okay? The ribosome is responsible for synthesizing proteins by creating these peptide bonds. So over here where we have the growing polypeptide, where these little black lines are attaching these different shapes, each of these black lines represents a peptide bond, which the ribosome creates. Okay, translation. Just like transcription, translation is a three-step process, okay? And the three steps go by the same name: initiation, elongation, and termination. Okay, now what happens during these three steps is where the differences come in. Initiation, okay, I break it up into steps. The first step of translation, called initiation, begins with mRNA at its 5' cap being attached to the small ribosomal subunit. Second, an initiator, tRNA, okay, with the amino acid MET for methionine, will attach to the mRNA start codon. Okay? So if you look here, they're showing you this here is this here. This initiator tRNA will have an anticodon that reads left to right UAC. And I already told you mmRNA, the start codon, is AUG. So the mRNA start codon, called AUG, okay, the base is AUG, will base pair with the anticodons, UAC. That ensures, by base pairing together here, which is here, that ensures the correct tRNA has transferred the correct amino acid. Well, AUG is not only a start codon, but it codes for the amino acid MET, which is methionine, and it's the only codon for this amino acid called MET. Okay. Third, what will happen is the large ribosomal subunit will then attach to the small ribosomal subunit, resulting in the initiator tRNA bound to the mRNA start codon being positioned in the P site. Overall, this results in the formation of the translation initiation complex. So overall, this results in the formation of the translation initiation complex. Okay. Now keep in mind, I said the start codon is closer to the 5' cap, okay? Another thing, in your notes, please make sure you note, I put it under mRNA. Not only did I give you a few codons you're responsible for, and you're only responsible for the codons I gave you in your notes, not that whole table I showed you, okay? So there's one start codon, 3 stop codons, and 2 codons I gave you, I believe, for tyrosine. But another thing I need you to know is that a single mRNA may have more than one codon, AUG. So what ensures, okay, that the ribosome knows which of those AUG codons is a start codon? It's in your notes. This codon AUG is part of a larger mRNA sequence called the Kozak sequence, Kozak sequence. It's in your notes and it's highlighted. If you look at that Kozak sequence, okay, you can see embedded or within it is AUG. So just underline the AUG and then above that put start codon. Okay, so that's what further helps the ribosome, okay, position the correct start, or helps the initiator tRNA and the ribosome locate which is actually the AUG that serves as a start codon. So here's our translation initiation complex. Once this initiation complex has been formed, the first step of translation is completed. Now we move on to the second step. The second step of translation is called elongation. Simply put, elongation is synthesis of the protein or synthesis of the polypeptide. You can just say synthesis of the protein if you want. What'll happen now, okay, is if you recall, I said the initiator tRNA is attached to the start codon and they're positioned in the P site. Why? So that the A site is accessible for the next tRNA. So what'll happen is when this initiation complex is formed, this initiated tRNA is in the P site, and then what I failed to point out is the next codon is now positioned in the A site. Okay, so it's ready for another tRNA to enter. So what will happen during elongation is the A site is available for the next tRNA to enter the A site. Now, which tRNA enters the A site depends on which tRNA has an anticodon that complements the mRNA codon. Okay? The correct tRNA will have an anticodon that complements the mRNA codon, and thus they will base pair together. That ensures that this is the tRNA that transferred the correct amino acid to the ribosome in the A site. Next, what will happen is the ribosome will take the growing polypeptide in the P site and it will chemically remove it from that tRNA in the P site and transfer that growing protein to the amino acid on the tRNA in the A site. And then what will happen, it says translocation, but what happens is the ribosome will shift, the overall ribosome will shift towards the 3' end of the mRNA. Except when the ribosome moves and shifts, it shifts and moves to the next codon, which they show you over here. But when the ribosome shifts to the next codon or translocates to the next codon, what happens is that takes this tRNA that's in the P site, okay, that's had the polypeptide or had the protein removed from it. It takes this tRNA and it moves it over to the E site where it can exit without any amino acids attached. Also, when it shifts, that causes this tRNA, which now has the growing polypeptide attached to it, that shifts this tRNA over to the P site, which they show you here, and now the A site is available for the next tRNA. This will continue to happen. That's why they show you it in a cyclical manner, in a cycle. This will continue to happen. As the ribosome shifts from one codon to the next codon to the next codon to the next codon, tRNAs will continue to enter. And what ensures the correct tRNA transfers the correct amino acid? Only the tRNA with an anticodon that complements the codon in the A site will be successful in base pairing and thus transferring the correct amino acid. But eventually what happens, as the ribosome continues to shift from codon to codon towards the 3' end of the mRNA, eventually what will happen is any one of 3 stop codons. Remember, not all 3. You won't find all 3 in one molecule of mRNA. You will only find one out of these 3 in mRNA. But eventually what happens is the ribosome will shift down to a codon And that codon will read either UAG, or it will read UAA, or it'll read UGA. Doesn't matter which one it is, okay? All three are called stop codons. First of all, there are no transfer RNAs with an anticodon that complements any of these stop codons. So that ensures that no more tRNAs transfer any more amino acids. Second of all, there is a protein called release factor. It very popularly in the sciences goes by RF. Okay. Release factor is the only protein and the only molecule that recognizes and binds to any one of these three stop codons. So notice, when the ribosome shifts down to the next codon along the mRNA molecule, and that next codon is a stop codon, no tRNAs recognize this, so none of them can base pair. However, release factor, who's been waiting around, waiting for his turn, okay, sees a stop codon go into the A site on the ribosome, and release factor will jump in. It's a protein, not tRNA, it's a protein. It'll jump in, it will bind to the stop codon, triggering the ribosome to release the polypeptide or protein, if you want to call it. Then it will trigger next the disassembly of the translation initiation complex. Everything eventually falls apart. Okay, so again, when a stop codon enters the A site, there are no tRNAs that have an anticodon that recognizes and binds to a stop codon. Only a protein called release factor recognizes and binds to stop codons. And when it recognizes and binds to a stop codon in the A site of the ribosome, it triggers the release of the polypeptide or protein and the disassembly of the translation initiation complex, thus terminating translation. Okay? So again, for both, gene expression is a two-step process, transcription, translation, but both processes, there has to be something indicating this is where you begin the process, and there has to be something indicating this is where you end the process. What scientists discovered, okay, is that technically what is being synthesized is a polypeptide, but for y'all, you can just think of it as proteins, okay? But what scientists discovered is just like the molecule of mRNA has to be made correctly, there can't be any mistakes. It's the same thing when the protein's being synthesized by the ribosome. There cannot be any mistakes, or else we can end up with a protein that's non-functional, or it's not behaving in the correct manner. It's not functioning correctly. Okay? I don't know about y'all, but I've been getting a heck of a lot of these scam calls. Okay, the last two things I want to cover here is just like after transcription in eukaryotes where pre-mRNA is synthesized and it has to go through post-transcriptional modifications, it's the same thing for proteins, okay? Depending on where the protein is going to do its work, some proteins have to undergo post-translational modifications. And I need to edit these PowerPoints to make it stand out. Okay, and I'll do that. There are two types of post-translational modifications that proteins can undergo. One is called glycosylation. This is where carbohydrates, or simply sugars, are attached to a protein. Okay, this is after gene expression is completed. In other words, translation is complete. We will have post-translational modifications. One of them is glycosylation. Glycosylation occurs in the rough ER, RER, and in the Golgi apparatus. Okay, in both of those membranous organelles, carbohydrates, or simply sugars, are attached to the proteins. For these proteins that have carbohydrates or sugars attached to them, often these proteins are going to be facing the extracellular environment, or they're going to be released into the extracellular environment. So one of the purposes for glycosylation, okay, is to attach a carbohydrate or a sugar to a protein so that this carbohydrate or sugar serves as an ID card for your immune system. So that when your white blood cells come across a protein that's been secreted or exocytosed by a cell, this sugar or carbohydrate that's been attached to the protein through glycosylation The protein can say, hey, here, look at my carbohydrate or sugar. And the white blood cells will say, okay, you belong here, go take care of your job. Okay? But another process occurs, and it has to do with protein folding. After gene expression is completed, after the second step translation is completed, not only may glycosylation occur, okay, but the formation of disulfide bonds may occur. When disulfide bonds are formed, they contribute to a protein's ability to fold. Therefore, they contribute to a protein shape. And if you recall, I said a protein shape is directly related to its function. Now, disulfide bonds are covalent bonds. Here's a disulfide bond. Here's a disulfide bond down here. Here are some down here. Here are some here. These are all disulfide bonds, okay? First of all, a disulfide bond is a covalent bond formed between the atoms sulfur. Okay, so the di refers to two atoms of sulfur, thus the di and the sulfide, and then the bond is a covalent bond created. What they're not showing you is this. There's an amino acid called cysteine. You can know it as cis. And the amino acid cysteine has a functional group. We talked about functional groups before, such as an OH group called a hydroxyl group, or a phosphate group that contributes to the formation of nucleotides. Those are functional groups. Well, this amino acid called cysteine has a functional group called a sulfhydryl group, or simply represented SH. Okay? And what was discovered is that when you look at a protein, okay, let's say it's this one over here, this line here, this gold line here, represents a chain of amino acids that are chemically bonded together through peptide bonds, except they just draw it as a straight line. Where you see the disulfide bond being formed, well, that means that there's an amino acid cysteine here, and there's an amino acid cysteine here. They're just not making it stand out. And what happens okay, is primarily in the rough ER. What will happen is there are enzymes that will locate the amino acid cysteines, okay, two of them, and if they're close enough together, and if it's required, what they'll do is they'll remove the hydrogen, freeing up electrons between the two atoms of sulfur, and they'll form a covalent bond. That's a disulfide bond. So for what I want you to know for post-translational modifications is that there are two types. Glycosylation, the addition of sugars or carbohydrates to proteins, serves as an ID card for the protein so that your white blood cells of your immune system know the protein belongs in you, allowing the protein to do its work. And then the second post-translational modification I want you to know is the formation of disulfide bonds between the amino acid cysteine. Know that a disulfide bond is a covalent bond between two atoms of sulfur, and then know the purpose of a disulfide bond. It contributes to protein folding, therefore contributes to a protein taking on the correct shape so it can do its job. Okay? I know it's a brutal chapter. It is. We're going to stop right there. Does anybody have any questions? I don't expect any questions, but does anybody have any? Okay, let me stop the recording. Listen carefully before you